are very late, I'm sorry. I'm gonna try to skip some stuff together. So I'm gonna kind of switch gears. I'm gonna tell you more about what we have done with the tools people like you or yourselves have developed, and how we have used these tools to answer an evolutionary question. And so this is work I did with Tatiana and Michael together as supervisors, and is the work from Marine and Fantan, previous PhD students, and a previous postdoc in the lab. Okay, so first, the biology. So we are dealing here with suppression of recombination. And so the first thing that you need to learn is that recombination is great, and it's because it allows, whenever you have, so whenever the parents met, they shuffle, so the offspring shuffles the chromosomes, and if these parents have different different sets of advantages, or at least not deleterious mutations, some of the offsprings, actually 1/4 of the offsprings, will have let's say a set of chromosomes that they can inherit afterwards. That is actually less. It has a lower load in deleterious mutations. But as recombination is great, is not always great, and this happened when you need any kind of locus in the genome, any loci, that needs to be in a permanently heterozygous state. And this happens notably in sex chromosomes. In that case, you cannot recombine, because at some point you can lose one of the alleles that is a sex-determining gene. And so at some point you ended up without sexes. So that's the suppression of recombination, and we know it's important. It has happened many, many, many times, and this is the origin of sex chromosomes. And the sex chromosomes, they came all in different flavors, and what is showing here, if you try to follow in that clockwise from a pair of autosomal genes, chromosomes, one of them acquires a sex-determining locus, and there are some species that still around that they have a single gene, actually a single SNP that determines the gene. This is like the case of the pufferfish. Some other species have but then things, when you stop recombining, things start going on, that they can go wrong, because any mutation that is linked to the non-recombining site is going to be also segregated with the heterozygous site. And so this process happens and goes on and on until you ended up with some examples, like they have lost entirely their Y chromosome, such as the spiny rat, or they had actually just need to acquire and form a new sex chromosomes, because the original sex chromosomes has degenerated so much. So this forms kind of a racket that cannot. So once you stop recombining, you cannot really go back. And so this is the idea is we are studying sex chromosome evolution. We know a lot of things. We know that sex chromosomes appears many times. We know that they appear because having this permanent heterozygous size is selected to allow sexual reproduction. We actually know a little bit how cool this has progressed. There are many competing hypotheses, but what we know a little bit less is when this recombination suppression start. And so I'm going to talk about dating the suppression of recombination events and what has happened then after. And I'm going to talk about transposable elements and how they accumulate in non-recombining regions. The degeneration of sequences, in sense of non-synonymous substitutions and optimal codon usage, gene losses, and then I'm going to put these two things together, and I'm going to tell you what has happened as a function and when it starts happening. And I call that the tempo. And for that, I'm going to switch for a model system that is ideal for testing this kind of hypothesis, because sex chromosomes have evolved there many times independently. So I have this kind of phylogenetically independent contrast methods that I can use. And in this case, I'm talking about plant castrating fungi that are called Mycobotrium. And in this case, they have, in this fungi, you have two different reproductive systems. One that is, we call a tetrapolar, in which the sex-determining gene, two sex-determining genes are placed in two different chromosomes. That means that after meiotic segregation, you ended up with 4 different types of gametes. On the other hand, for some other species, you have a bipolar system in which the two sex-determining genes, they are put together in the same chromosome. That means that after meiotic segregation, you ended up with only 2 types of gametes, and with the advantages that after the meiotic segregation, you have half of the progeny, half of the gametes are compatible with you, while in the case of tetrapolar species, it's only 1 out of the 3 other gametes that were produced. So this bipolarity is advantageous in high sulfurs, and this is exactly what these species are. And so we know this independent evolution. I'm going to skip this over. There are multiple transitions, because some of the species, the tetrapolar species, are scattered all along the tree. So that means they had retained the bipolar, the tetrapolar ancestral state, but others had actually fused the two ancestral, so the two chromosomes that have the sex-determining genes, they had fused together in a single chromosome. We know that this has happened independently, mainly beginning by checking out what is the ancestor, the combination of the sex chromosomes, and in this case, 2 different types of rearrangements has appeared. One in the second species, and another one in the 3 bottom species. We know they are different. We do not know if the 3 species in the bottom are actually independent evolution of sex chromosomes, or they are sharing a single scenario. And for that, I went to developing a tool that is called a phylogenetic scan. And what I'm showing here is, so just if you follow these synteny tracks, you will see, I have a pointer, awesome. If you follow these synteny tracks, the ancestral state is these 2 different chromosomes. You'll see that in the bipolar species, one part of this chromosome has become an autosome. That means it's a fully recombining chromosome, but the other part has fused with the entire chromosome. In this case, it's called PR, and now it's not recombining, right? So when I do this phylogenetic scan, this green part that is now autosomal, I will expect this every single gene, or in this part, is actually following the same topology as the species phylogeny, because they are just segregating as any other autosome. And this is actually what I found. I did this by ultrametric trees. I'm going to skip the details. You can ask me after. Most of this is published. So, and then I go. I'm going to go to the different parts of the chromosomes, and in this case, you see that the species tree and the chromosomal sex chromosomes tree is completely different. And you have, in particular, this kind of what we call a trans-specific polymorphism, in which the sex chromosomes segregate by mating type rather than by species tree. And you go on, and then some of them keep going trans-specific, but then some others remain in their separation by species, until you reach a point in which you have again the species tree, but then the branch lengths are very different. We have done from plenty species, we have identified more than 20 different onsets of suppression of recombination, but this analyzing gene trees and testing for the coherence with the species tree. And with that, we know when, so more than 20 times recombination has stopped in this species. Now, what has happened since? And first, Marine, former PhD student, developed this pipeline for annotating genomes and identify transposable elements. Is actually just a bunch of other tools put together, and then more phylogenetically aware way of annotating the transposable elements. And what she found is that transposable elements has accumulated like maybe 10 times as much. They are more. They constitute a fraction of the sex chromosome that is almost 10 times larger than what they have in the recombining chromosomes, in the autosomes. And this is for all kinds of transposable elements, but mainly for long non-terminal repeats. Then another former PhD student in the lab, he studied the accumulation of sequence degeneration signals. We did that like with normal Shihen-Zhang's GN00 BNDS calculations, but we also check whether the genes in the recombining and the non-recombining regions, do they have a different codon usage? And actually, they do. The fraction of optimal codons used by the non-recombining region, so the sex chromosomes genes, is lower than in the rest of the genome. Then went on, look at the number of genes that are still present in the sex chromosomes, and how many of those genes that they are supposed to have to be in 2 copies in the 2 sex chromosomes, how many times they have now 1 copy lost. We call these hemizygous genes. And he found that roughly between 5 and 50% of the genes, now they became hemizygous. But she also found that roughly 5 to 50% of the genes in the sex chromosomes, they do not exist in the tetrapolar species. So there are a lot of new genes being created. And so now we are going to dance. We are going to put what happened as a function of the time since it happened. So first, the accumulation of transposable elements. You go a very sharp increase, and then it goes over a plateau that it saturates around 2 million years. The sequence degeneration, it's more like an instantaneous decrease in the number of optimal codons used by sex chromosome genes, and then also reaches a plateau, and this plateau is above the expectance of using only the optimal codons. And there is a lot of asymmetrical gene losses. So one gene can be lost in one sex and present in the other, and one gene in the other will be lost in the first. And so what I just say is that the suppression of recombination can be accomplished in a stepwise manner. This is the analysis of the gene trees I showed you at the beginning. Transposable elements and sequence degeneration saturates, but at different timing or with different dynamics. And gene losses continues the appearance of new genes. And so if I put everything together, if you go from this ancestral state, you fuse your 2 chromosomes, you put the 2 sex-determining genes together, and then immediately sequence degeneration kicks in. So you start accumulating substitutions, and these substitutions reach a threshold, like if there is a mutational load that cannot go further, that sex chromosomes cannot go further, maybe because of selection. But then transposable elements start accumulate. And sometimes they will actually be inserted in the middle of a gene, and that gene it disappeared. And once you have enough transposable elements, these are transposable, they can actually go under non-homologous recombination, and so they can produce structural variants, so inversions and large-scale deletions. This is what I'm showing here. These two transposable elements recently, they just flip, and now you have a different gene order in the extant chromosome, which means, perfect. Which means that you have now a completely rearranged sex chromosomes. The two chromosomes they are doing this independently because they are not recombining, and this is also growing the size of the non-recombining regions. But now you have a nearly linear asymmetrical gene losses, but also new genes are formed. And some of those new genes are maybe just a transposable element inserted in or nearby a gene that it's just the transposable element is kind of domesticated by the genome and then stays there. So the 5 to 10% of new genes that I told you before, these are genes that are conserved across the species. So they appear and then many of them remain. And with that, I'd like to finish. Thank you to all my lab, the co-head of the lab, that is Tatiana Giraud, and my students, my postdocs, and to you for listening. Thank you very much.
We have time for a couple of questions. Any questions from audience?
I can try shouting.
I repeat the question. I cannot see you because I have the lights here, so it's somewhere over there.
Yeah.
Yes. Hello. We met yesterday.
We did. What's the driver of recombination inhibition? Is it the inversions, or is there some other mechanism that inhibits the recombination?
So inversion is an obvious, because once you are inverted, you cannot implement it, you cannot pair. But there are some other mechanisms, notably if the transposable element load is different. So actually you look synthetic, but you are not, because you have big differences. And then we have another piece of evidence, and I didn't have time to tell you about, is the DNA methylation. Since these non-recombining regions are full of transposable elements, the first thing the genomes try to do is to inactivate those transposable elements by methylating the stuff. And methylation itself reduces recombination rates. And if it happens nearby a place that is already non-recombining, it can be captured as a non-recombining, and the non-recombining regions will extend. And yes.
Is the recombination inhibited equally across the chromosome, or so that are the recombination hotspots in particular?
Yeah, good question. And yes, so it's different because the recombination, the region that is suppressing recombination, it extended in a stepwise manner. And so in the first event of recombination suppression, they have no recombination, completely arranged. But then you have the next region that was incorporated. It has a little bit less rearrangements, a little bit less transposable elements load, a little bit less sequence degeneration. So yes, there are differences, and these differences are actually the things we use to identify the stages in the suppression of recombination. Yeah, good question. Yeah.
Okay, another one more. Oh,
I'll just shout this.
Yes, I try to.
The transposable element data you showed for autosomes, pseudoautosomal region, and non-recombining, the pseudoautosomal regions look to me somewhat different. Was that significant?
Yes, actually, great point. You were watching the slides. Awesome. And so if you see, so recombining regions to the left, non-recombining to the right, and in the middle, I didn't say it is pseudo-autosomal region. This is the region that still recombines in sex chromosomes. And these are actually just flanking the non-recombining region. I skipped for the sake of time. And if you see, and you need to believe me because the stats are not there, but most of the curves, most of the line, they start like low, then go lower, and then explode. So it's like autosomal recombinant average, then smaller, and then just crazy. And this is real for non-terminal LTRs. Not so much for elytrons, but these are a different kind of, and anyway, you have very little to begin with. So why there are smaller, less transposable elements in pseudo-autosomal regions? And that's a great question. And my answer, I think, is because since these guys, they do not recombine over the non-recombining region, but they still need to recombine. Otherwise, they cannot segregate, right? So, and since these regions are rather small, the average number of recombination events per base pair holds as in the rest of the genome, but it holds for a smaller region. So they recombine more than autosomal. And that's the entire, that's my answer.
Thanks.
Thank you very much.